In the world of microbiology, a unique class of infectious agents exists that defies traditional classification. Acellular microorganisms are infectious agents that lack a cellular structure. Unlike bacteria, fungi, and parasites, acellular microorganisms lack the fundamental components of cells. They don't have cell membranes, organelles, or the ability to metabolize nutrients. This makes them fundamentally different from typical microorganisms. There are three main types of acellular microorganisms: viruses, viroids, and prions. Viruses consist of genetic material, either DNA or RNA, encased in a protein coat called a capsid. Some viruses also have an outer envelope. Viroids are much simpler. They are small, circular RNA molecules without a protein coat. They primarily infect plants. Prions are the most unusual. They are simply misfolded proteins without any genetic material at all, yet they can cause fatal neurodegenerative diseases. What makes acellular microorganisms unique is their complete dependence on host cells. They cannot metabolize nutrients or reproduce on their own. Instead, they must hijack the machinery of a host cell. After using the host's resources to replicate, new infectious particles are released, often destroying the host cell in the process. To summarize, acellular microorganisms have 5 key characteristics. They lack cellular structure, cannot reproduce independently, cannot metabolize, rely completely on host machinery, and include viruses, viroids, and prions. Acellular microorganisms cannot replicate independently due to their fundamental lack of cellular machinery. Unlike cellular organisms, viruses, viroids, and prions must hijack the host cell's molecular machinery to reproduce. Viruses are composed of genetic material, either DNA or RNA, surrounded by a protein coat. The virus replication cycle involves several key steps. First, the virus attaches to specific receptors on the host cell surface and enters the cell. Inside the cell, the viral genetic material is released and uses the host's enzymes and resources to replicate its genome. The host cell's ribosomes then synthesize viral proteins using the instructions from the viral genome. These viral components then assemble into new virus particles inside the cell. Finally, the new viruses are released from the cell, often causing cell lysis or death, and can go on to infect other cells. Viroids are significantly simpler than viruses. They consist of a small, circular RNA molecule without a protein coat. Viroid replication follows several distinct steps. Viroids enter plant cells through mechanical wounds or during plant propagation. Once inside, they migrate to the nucleus where plant RNA polymerases recognize their structure. Unlike viruses, viroids do not encode any proteins. Instead, they hijack the plant's RNA polymerase to catalyze a rolling circle mechanism of replication. The plant's own RNA processing enzymes then cleave the long RNA strands into individual viroid genomes and circle them. These newly formed viroids can move through the plant's vascular system, spreading the infection to other parts of the plant. Prions represent a unique mechanism of replication that does not involve genetic material at all. Unlike viruses and viroids, prions replicate through a process of protein misfolding and aggregation. First, a misfolded prion protein enters the cell or forms spontaneously through mutation. This abnormal protein comes into contact with normally folded proteins of the same type. The prion then induces the normal protein to change its conformation and become misfolded as well. This process creates a cascading effect where each misfolded protein can convert more normal proteins. Eventually, these misfolded proteins aggregate into insoluble amyloid fibrils that disrupt cellular function and lead to neurodegeneration. To summarize, all acellular microorganisms depend on host cells, but each has evolved a unique strategy for replication. Despite their structural and functional differences, viruses, viroids, and prions all share the fundamental characteristic of being entirely dependent on host cells for their propagation. HIV, the human immunodeficiency virus, is a retrovirus with an RNA genome. It primarily attacks and destroys CD4 plus T cells of the immune system, leading to AIDS if left untreated. HIV is transmitted through direct contact with bodily fluids such as blood, semen, or breast milk. The influenza virus has a segmented RNA genome that contributes to its high mutation rate, a phenomenon known as antigenic drift. This explains why we need new flu vaccines each year. Influenza causes seasonal flu epidemics and is primarily transmitted through respiratory droplets when an infected person coughs or sneezes. SARS-CoV-2 is a coronavirus with an RNA genome and distinctive spike proteins that give the virus its crown-like appearance. It emerged in late 2019 and caused the global COVID-19 pandemic. Like influenza, it spreads primarily through respiratory droplets and aerosols. These three viruses have had significant global health impacts. HIV continues to affect millions worldwide, with approximately 680,000 annual AIDS-related deaths. Seasonal influenza claims between 290,000 and 650,000 lives each year. SARS-CoV-2 has caused approximately 7 million deaths since the pandemic began. Viroids are small, circular RNA pathogens that primarily affect plants. Unlike viruses, they lack a protein coat. Their structure consists simply of a circular RNA molecule that can fold into a complex shape. The potato spindle tuber viroid, discovered in 1971, causes a serious disease in potato plants. Infected potatoes develop an elongated spindle shape compared to healthy round potatoes. Symptoms include elongated tubers, stunted plant growth, and significantly reduced crop yields of up to 40%. The tomato planta macho viroid affects tomato plants worldwide. Infected tomato plants show distinct differences from healthy plants, with notable yellowing and reduced size. Symptoms include loss of chlorophyll, stunted growth, reduced fruit size, and leaf deformation. The avocado sunblotch viroid affects the quality and quantity of avocado production worldwide. Infected avocados develop characteristic yellow or red streaking on the skin, called sunblotch. Symptoms include distinctive yellow streaking, smaller deformed fruits, and reduced yields of 15 to 30%. These viroid infections collectively result in millions of dollars in crop losses annually, affecting agricultural economies worldwide. The classification of living organisms follows the three-domain system: bacteria, archaea, and eukarya. Acellular microorganisms like viruses, viroids, and prions don't fit into this classification system. These entities are excluded from the tree of life because they lack cellular structure and cannot perform independent life processes. Their classification as living or non-living entities remains a matter of scientific debate. Scientists continue to study these entities to better understand their place in biology and the broader context of what we define as life.